Hi, in this video we're going to talk about the process of translation and post-translational modifications that will occur to a polypeptide before it becomes a fully functioning protein. So first, let's talk about the machinery that's involved in translation. Now we know that our mRNA transcript is going to provide the code for how to build the protein, but we need the machinery, or the factory, so to speak, that's going to actually churn out that protein and read that code. So translation is carried out both by ribosomes and tRNA. And again, it's going to occur in three stages: initiation, elongation, and termination. Those same three stages that we see coming back again and again. So let's start by talking about tRNA, or transfer RNA. Now, tRNA binds to codons of the mRNA, and it matches the appropriate amino acid to that codon. So, first, how does tRNA bind to a codon? Well, it does this with an anticodon, which is a complementary code to the codon. So here we see the primary structure of tRNA, which is simply the sequence of bases. And you can see that right here, this region, we have the anticodon loop, and that is going to be what binds to the codon on mRNA. Now, we also have to have a way of getting amino acids onto this tRNA, and it turns out that this 3' end is the attachment site for amino acids, and this particular tRNA here happens to be the tRNA that carries phenylalanine, and that's this amino acid right here. So here we have phenylalanine. Don't worry about the name too much. Again, you don't really need to know the names of amino acids, but just for completion's sake, that's phenylalanine, and that is going to attach here at this 3' end, which is the attachment site for amino acids. Now, tRNA actually has secondary structure that we see right here. So, color-coded are the different sections of the tRNA structure. Like you can see here, this models the shape we see over here. It just doesn't have the actual base sequence written in. But these colors will help you visualize how this shape turns into this shape, how it twists around, and where the different components come from. So here, this portion right here, that is our anticodon. And here, that's the amino acid attachment site. And this enzyme, amino acetyl tRNA synthetase, that's the enzyme that catalyzes the transfer of an amino acid to the tRNA. Now, let's talk a little bit more about that codon-anticodon binding, because it turns out we haven't talked about the whole story yet. So you might remember that there are 61 potential, or rather 61 potential codons for amino acids, right? There would be 64, but 3 of those codons are stop codons and don't actually code for an amino acid. So we have 61 codons. That means that there would be 61 potential anticodons, or 61 different types of tRNA that could be found in a cell. However, most cells only have about 40 different types of tRNA. So, how can we have all these codons, these 61 different codons, but only 40 tRNAs to translate them? The answer is something called the Wobble Hypothesis, which basically says that there's some unconventional base pairing in the third base of the codon anticodon binding. So let's say that our codon is UC. That is going to be our codon, which happens to be the codon for phenylalanine. And our anticodon for that would be AG. And there we will have our conventional base pairing, 2 between the A's and the U's, and 3 hydrogen bonds forming between the G's and the C's. What the Wobble hypothesis is all about, though, is the fact that, let's say that in our tRNA, that the only tRNA for phenylalanine we have is actually one that has an anticodon of A. So we know that UC is our codon, right? Here, that's our codon. And now our anticodon is no longer a perfect match. These first 2 bases are still fine, right? We can still form 2 hydrogen bonds between those U's and A's. But normally we have a G hydrogen bonding to a C, not an A. However, we can still have a match between this codon and anticodon because of Wobble, because we can see some unconventional base pairing between that A and C. It's not a perfect fit, but it'll still work here. So that is the Wobble hypothesis, that third base of the codon anticodon can actually have an imperfect match to what the base is on the codon. So that means that we don't need a tRNA for every single codon there is, that some tRNA will be flexible and be able to bind to more than one codon. Now, let's flip the page and talk about ribosomes.